there is one topic about Lord Voldemort that I haven't went into too much detail on. Oh, what's that? YouTube is relaxing demonetization on this topic? Well then, there's no need for me to keep avoiding the word coronavirus anymore. So, as you know, there are a lot of conspiracy theories going around. The biggest one is probably the idea that this virus was man-made as a bioweapon of some sort from China. And you know, this isn't the first time that China has kept something secret, so it's not entirely impossible, right? Well, the thing is, we don't need any information directly from China to know that this virus is not man-made. A while ago, I stumbled upon this video by Fizzy Onic, which was uploaded a month ago. He's a PhD student in molecular medicine, so frankly, he's a lot more qualified to speak about this topic than I am. But I thought we could show him some love since he is a small channel that makes a lot of videos about science and biology, which is great, but they don't have many views. If you're interested in learning about molecular biology, I recommend checking his channel out. Now, how did I find this guy? Well, one of his videos went semi-viral, and it's about if the coronavirus was man-made or not. In his comment section, he seems to have attracted a lot of these conspiracy theorists. So I'm going to play a few clips of his video because frankly there was some information in there that I wasn't even aware of. But before we get to that, I must mention today's sponsor, NordVPN. You've heard of them, right? Of course you have. There's no way you haven't heard of them. Nord is an encryption service that hosts over 5,500 servers in 60 countries. No matter what you need a VPN for, Nord is the way to go. No matter if you're trying to unlock Netflix while you're stuck overseas or just to protect your data on public connections. You can connect to 6 devices with only one subscription on any of the major operating systems including PC, Mac, Linux, Android, and iOS. If you're interested in protecting yourself while surfing the web, be sure to head over to nordvpn.org/stick to get a deal of 70% off the 3-year plan. Or, of course, you can just use the code STICK. That's nordvpn.org/stick. Thank you to Nord for sponsoring this video. Now, without further ado, let's have a look at Fizzy Onic's video. We're going to learn something today.
Hi, my name's Nick. I'm a PhD student in molecular medicine, and in this piece of content, I'm going to briefly answer if it is possible that coronavirus, or COVID-19, is a man-made virus based on a recent review published in Nature. And that's a highly reputable biology journal, not Trees. We can answer the question of if this virus is man-made or not by looking at a few clues. The coronavirus is made up of proteins, just like any other biological thing, and a specific piece of that virus known as a spike protein with a receptor binding domain that is used to attach to a receptor found on human cells, known as angiotensin converting enzyme, or ACE.
Just to add a bit more detail, viruses will always be attaching to a specific receptor on the host cell. It's this attachment mechanism that ultimately signals the virus to inject its genetic information into the host. But like everything, these receptors and attachment proteins are specific. If they target a protein found in humans, for example, then the virus likely infects humans. Basically, the virus infects whatever type of organism their proteins are specific to. Let's say a virus infects some closely related animals to humans, say chimps, or even slightly more distant ones like bats. At first, they don't necessarily infect humans, but once they undergo a crucial mutation in their outer proteins to make them specific to a human cell receptor, then that's a way a virus can jump from an animal to a human. Pretty simple, right? Of course, a few other conditions would also have to be met, but we won't get to that right now. COVID-19 specifically targets a protein found on human cell membranes called ACE2, and ACE2 is most commonly found in alveoli cells within your lungs. That's why the coronavirus mostly attacks your lungs. Depending on what the virus targets, it's going to affect different tissues differently, and SARS-CoV-2 is no exception. Now, you're probably thinking, what is this ACE2 receptor that's there just for the virus to infect me? Well, these proteins already have existing other functions. ACE2, for example, participates in a mechanism that lowers blood pressure, which is part of a negative feedback loop. The SARS-CoV-2 virus takes advantage of this and uses it as a signal to infect the cell. Anyway, that was just some random information. Let's carry on.
That receptor binding domain is made up of a series of amino acids, just like all proteins are made of amino acids. This receptor binding domain is made up of 6 amino acids, of which 5 are different from the first coronavirus to induce panic in the world. This receptor binding domain has a high affinity for the ACE receptor in human cells, meaning it really likes the receptor and will bind to it often. However, its interaction with the ACE receptor is characterized as not ideal, giving us a clue that it might bind something else better, maybe something non-human. If this were a purposefully created biological weapon, you'd imagine it would be created with an ideal receptor binding domain to be more effective and thereby deadlier.
Yes, a very good assumption to make. A zoonotic source of the virus would mean it isn't exactly perfect. Let's take a look from the original source. Mutations in the receptor binding domain of SARS-CoV-2. The receptor binding domain in the spike protein is the most variable part of the coronavirus genome. 6 RBD amino acids have been shown to be critical for binding to ACE2 receptors and for determining the host range of SARS-CoV-2-like viruses. With coordinates based on SARS-CoV-2, they are blah blah blah. 5 of these 6 residues differ between SARS-CoV-2 and SARS-CoV. On the basis of structural studies and biochemical experiments, SARS-CoV-2 seems to have an RBD that binds with high affinity to ACE2 from humans, ferrets, cats, and other species with high receptor homology. While the analysis above suggests that SARS-CoV-2 may bind human ACE2 with high affinity, computational analyses predict that the interaction is not ideal and that the RBD sequence is different from those shown in SARS-CoV to be optimal for receptor binding. Thus, the high affinity binding of the SARS-CoV-2 spike protein to human ACE2 is most likely the result of natural selection on a human or human-like ACE2 that permits another optimal binding solution to arise. This is strong evidence that SARS-CoV-2 is not the product of purposeful manipulation. Now, we can even go into the original source that made this analysis. Several other critical residues in 2019 nCoV RBM, particularly asparagine 501, are compatible with but not ideal for binding human ACE2, suggesting that 2019 nCoV has acquired some capacity for human-to-human transmission. You can actually go further down past the abstract to see some of their explanations for protein structural analyses, multiple alignments, and phylogeny tree depictions. But anyway, this isn't the only evidence we have. What else is there? Well, it seems that the Daily Situation Report by the WHO has made a few statements on this. As I'm recording this, it's April 24, and the WHO had this to say. All available evidence for COVID-19 suggests that SARS-CoV-2 has a zoonotic source. Many researchers have been able to look at the genomic features of SARS-CoV-2 and have found that the evidence does not support that SARS-CoV-2 is a laboratory construct. A constructed virus would show a mix of known elements within genomic sequences. This is not the case. SARS-CoV-2 was identified in early January and its genetic sequence shared publicly on the 11th and 12th of January. The full genetic sequence of SARS-CoV-2 from early human cases and the sequences of many other viruses isolated from human cases from China and all over the world since then shows that SARS-CoV-2 has an ecological origin in bat populations. All available evidence to date suggests that the virus has a natural animal origin and is not a manipulated or constructed virus. Many researchers have been able to look at the genomic features of SARS-CoV-2 and found that the evidence does not support that SARS-CoV-2 is a laboratory construct. If it were a constructed virus, its genomic sequence would show a mix of known elements. This is not the case. All available evidence for COVID-19 suggests that SARS-CoV-2 has a zoonotic source. Since there is usually limited close contact between humans and bats, it is more likely that transmission of the virus to humans happened through another animal species, one that is more likely to be handled by humans. This intermediate animal host or zoonotic source could be a domestic animal, a wild animal, or a domesticated wild animal, and as of yet has not been identified. Anyway, what's the takeaway here? Okay, so when we try to create something new in a laboratory, there's usually little to no room for invention from scratch. For example, let's say we're trying to develop a new antibiotic. If that's the case, scientists don't create a new molecule from scratch. Usually it's done by looking at things that already exist in nature. For example, many fungi produce antibiotics, and we use those to create new drugs. This isn't just the case for medicine, but is also the case for many other things too, including genetic manipulation. If you've ever seen Jurassic Park, remember how they mix in genes from other species in order to, fill in the gaps? Inspiration is usually taken from other parts of nature. Let's say you wanted to alter the genes of an embryo so that they have night vision or something. Just a wild example. You're not going to find this gene just by going to a laboratory, introducing some mutations, or creating a new gene from scratch. No, you would grab something that already exists in nature and make it compatible, maybe from a red fox, for example. I don't know. So in other words, if we're looking at the potential idea that the SARS-CoV-2 virus was created in a lab as a bioweapon, we would expect to see something like that. Its genetic code would have bits and pieces from other things in nature, perhaps from other viruses. That's what the WHO means when they said a mix of known elements. Do we see that in SARS-CoV-2? No. Feel free to look up the RNA sequence yourself if you're in doubt. It's been available in public databases since February. So there you have it. 2 strong pieces of evidence we have so far. If you have any friends or relatives who still think that China artificially created the virus as a bioweapon, feel free to show them this. Now, on the bottom of Fizzy Onic's video, there are some questionable comments, so let's quickly address them. They didn't want a virus that shows signs of a perfect biological weapon. They wanted a virus that does the job but still raises no suspicions. Of course, that is certainly within the realms of possibilities. If the scientists really took a lot of care into designing a virus, in theory they could create something like SARS-CoV-2 and hide it. However, that's just so improbable due to the difficulty of executing such a task that it's just nonsensical to even consider it as true. What do you think is more likely? I think it being natural and undergoing natural mutations to get to where it is now is much more probable than scientists designing it to look that way. Not to mention that artificially created bioweapons would still have some sort of known elements within genomic sequences due to the impossibility of creating functional genes from scratch. That violates highlights the second point I made. So is this idea impossible? No, but it sure as hell isn't likely in any sense. Anyway, that's my time. Thank you all for watching, and thank you to Fireshard, Liam, Don Jessica, Joe Goldenberg, and JN. YouTube's ad revenue has really been hit hard with COVID-19 going around, so it means a lot that you guys are still willing to support me through these difficult times. Thank you.